Welcome to the ASGCT 2026 annual meeting in Boston. This week we are talking with some of our ASGCT members to learn about their backgrounds and talk about ASGCT membership. I'm here today with Mark Deehan from MassGeneral Hospital. Mark, welcome
Hello.
to the show.
How are you?
I'm doing well.
Yeah.
Excellent. So we're just going to take you through a couple different questions here. We would love to learn a little bit more about your role and the work that you're focused on right now.
Yep. So at Mass General right now, I'm an instructor in neurology, and I did my postdoc in Marion DeFilia's lab, and so I'm continuing that work as an instructor, and so I was brought into her lab to study a neurodevelopmental disorder called NACC1, which stands for Nucleus Accumbens Associated 1, and it's an ultra-rare neurodevelopmental disorder. There's only about 50 or so families known worldwide, and we established the first knock-in model for the most prominent mutation in that gene for both mouse and human pluripotent stem cells. So we've been studying kind of the basic biology, like what that mutation might be causing in neurons or in brain to drive that disorder. So that's been most of my work to date.
Fantastic. Sounds good. And this is something I always love to ask. What first drew you into the cell therapy and gene therapy space? For example, like I initially went to university, I had grand plans of being an optometrist. And I found out I was too dumb to do that, but I did like biology, and so I studied that route, but then didn't actually get into gene therapy until the end of my master's. Yep. And here we are.
Yeah, I might be on a similar path there. So when I started in Marion's lab, she works a lot with Dr. Anastasia Korova and Dr. Neil Aronin at UMass Medical School to develop divalent siRNAs. So they have a very strong, therapeutic research avenue for Huntington's disease using those molecules. But my focus mainly was the basic biology of this new disorder. So, I was gene and cell therapy adjacent for a long time, and now I've recently taken more of a stab into that field, trying to get collaborators to help with that disorder and with Angelman's as well. So,
sounds good. What part of the space are you most excited about at the moment?
I think it's application, right? So it's really, getting into the clinic a lot more, and you have a lot more therapies that are being able to be generated faster and tailored to ultra rare and rare populations. And so I think that's, very important.
Sounds good. What challenge or opportunity in the field do you think deserves more attention?
So, for instance, being new to the field, I would say that, I focus a little bit more on kind of the research that I have done at that point. And the NACC1 gene is a transcription factor, for instance, and it's a neurodevelopmental disorder. So I'm very curious to see, when you generate these gene therapies and in preclinical models they work, very well, but not all of the phenotypes are able to be reversed. And that, might be the case in the clinic as well. So I'm curious to see how the field advances to see if there's any additional modifications or therapeutic avenues to try to reopen therapeutic windows that might have closed, especially in the neurodevelopmental space. So that's somewhere where I'm interested in personally, but I'm curious to see where it goes because it feels a little bit of an unmet need.
Excellent. And shifting gears here just a little bit, how has ASGCT membership helped you in your career?
So I'm a new member of ASGCT, but I would say I was luckily enough awarded the Career Development Award.
Congratulations.
Thank you very much. Yeah, in association with the Foundation for Angelman Syndrome Therapeutics. And so that's a ground-up project that we had started, about 4 or 6 months ago once we were administered the award. And so we're just getting cell lines in the lab up and running to test a divalent siRNA to try to knock down the paternal antisense strand and re-express the paternal UBE3A that's normally silenced in individuals with Angelman. So it's a very high-risk project. I don't think divalent siRNAs are well characterized in purely nuclear targets, so it's a pretty high risk, high reward project, and I'm excited to see where it goes.
Fantastic. And separate question, but somewhat related, why has ASGCT continued to be valuable to you as a member?
So I think the conferences are great. You get to show up and see a lot of science and meet new people. So that is something new to the gene and cell therapy field. It's just absorbing as much information as possible. And then I'm going to start to look into what online resources are offered too, because it seems like there's a lot that is offered that I don't think I've taken advantage of yet, but maybe I should. So I look forward to that as well.
Excellent. Well, thank you so much for coming on the show.
Thank you for having me. Yeah.